Hey, welcome back. Today we're taking a look at transport across the cell membrane. So starting off with what we call passive transport. Moving things across that cell membrane is one of the most important jobs that the cell membrane has, regulating what gets into the cell and what is able to leave the cell. There's two main types of transport. The first is passive transport. That's what we're going to talk about first. And then we'll get to active transport. As the word passive kind of implies, passive transport requires no cellular energy. Usually it's a form of diffusion, and it moves particles down the concentration gradient. So there's that phrase again. We talked about that a couple days ago when we did diffusion. We are moving things down the concentration gradient. So from high concentration to low concentration. We discussed diffusion, and there are some particles, some molecules that are able to diffuse straight through the cell membrane. They're small enough and uncharged, and so over time they're able to go from a high concentration to a low concentration, and then we have equal concentrations, and those particles can go straight through the cell membrane. For larger molecules, or charged particles that can't just squeeze between those phospholipids, we have another process called facilitated diffusion. And this involves the use of an integral protein. So remember, the integral proteins are proteins that span the entire phospholipid membrane. They have, go straight through it. They have a top and a bottom to them. The first type of facilitated diffusion happens through a protein called a channel protein. And we see that here on the bottom. Basically exactly what it sounds like. It's just a channel, and a molecule, we have a charged ion here, just flies right through it. There we go. There it is. The other type of protein is called a carrier protein. Carrier proteins change shape. So we have that illustrated here. It's open to the outside. The molecule goes in. Then it closes to the outside and opens to the inside, and the molecule spits into the inside. So here's our glucose carrier here for a large glucose molecule. And again, you watch it change shape as the molecule travels through it. So if it changes shape, it's a carrier protein. If it just has one single pathway through it, doesn't change shape, it's a channel protein. So both of these are called facilitated diffusion because they are diffusion. We're moving from high concentration to low concentration, down the concentration gradient, and it's a natural process. It requires no energy, but the word facilitated comes in because it's facilitated by a protein. It's helped by a protein. So facilitated diffusion is diffusion with the use of a protein, whether that be a carrier protein or a channel protein. So that was passive transport, and next up we have active transport. And the big difference between passive and active is that active transport does require energy. And generally the reason for that is because it's moving particles against the concentration gradient. So against, or up the concentration gradient, would mean it's moving particles from a low concentration to a high concentration. Cellular energy is usually in the form of ATP. So you see that here. We've got the starburst around there, kind of your universal symbol for energy. ATP is used to power this protein pushing particles against the concentration gradient, and that would be active transport. The proteins that do that pushing are called pumps, protein pumps. So these, like carrier proteins and channel proteins, are integral proteins. They span the entire membrane. Unlike the carrier and channel proteins, these push molecules against the concentration gradient. You see that here. They're moving them against concentration gradient. With a low concentration on the bottom and a high concentration on top, and we're moving things from low to high. That's against the concentration gradient. That's why it requires energy. And we see that here. Here's our energy molecule, ATP, and it's providing the energy to move those molecules from low concentration to high concentration. Very large molecules, some macromolecules, or a large quantity of molecules enter the cell through a different mechanism. and it usually uses what we call vesicles. So here's a picture of a vesicle. There's the GIF with the vesicle right there. There's a vesicle, that kind of inner purple circle. And what's going on here is something called endocytosis. So what happens is if the cell needs to engulf this large quantity of orange molecules, whatever they are, or a large green molecule that is way too big to pass through any kind of protein, what happens is that the cell kind of engulfs them, it surrounds them, and the cell membrane, this light blue color here, or the purple color here, pinches inward, and you see that happening. So here's step one, here's step two, that cell membrane is pinching inward, and eventually it's going to cleave off. And that process is called endocytosis. And now you have this new molecule, there's a whole group of molecules now inside the cell, kind of in their own little bubble there, and that bubble is called a vesicle. And it can travel to different parts of the cell and do different things, and we'll take a look at some different types of vesicles when we get to our cell organelle chapter, which is next. If the material taken in is solid, we call it phagocytosis, and if the material taken in is liquid, we call it pinocytosis. So just a couple more words there to keep you honest. Phagocytosis for solids, pinocytosis for liquids. And finally, we have exocytosis. This one sounds very similar to endocytosis, and indeed it is. It's basically just the opposite of endocytosis. And this exocytosis, a vesicle slowly moves towards the edge of the cell. The edges of it fuse with the cell membrane and actually become a part of the cell membrane, and everything gets pushed out into the extracellular space. So you can see that picture here. Here's the image. You've got the vesicle. It moves towards the cell membrane. It merges with the cell membrane and pushes all those particles out of the cell. So endocytosis is into the cell. Exocytosis is out of the cell, exiting the cell. Both endo and exocytosis require energy, and therefore they are forms of active transport. Remember that term endocytosis. We're going to come back to it in Unit 4, which is next, and we're going to talk about the endosymbiotic theory. So we'll see this again. Quick recap of the lesson here. We have two types of transport, passive and active. Passive requires no energy. Generally works by simple diffusion, and also facilitated diffusion, where you have the proteins that help the molecules move through. Facilitated diffusion is either a channel protein, so see that here, just kind of a protein that doesn't change shape, but lets molecules pass through, or it can be a protein carrier, where it does change shape. Active transport does require energy, and you're usually going to know that because you've got the ATP symbol. There's like a power symbol there. And you're also moving molecules from low concentration to high concentration. So the squares are of high concentration down here, low concentration up here, but we're moving against the concentration gradient. And then finally, we talked about endocytosis and exocytosis today. That's where we move large amounts of molecules, or large molecules themselves, either out of the cell, here's exocytosis, or into the cell, the endocytosis. So that's a recap of what we went over today. Hope that helps. I'll see you guys tomorrow.